Today I am taking up the topic translation. Translation is a part of gene expression. Gene expression has actually two parts: transcription and translation. Transcription takes place inside the nucleus and translation occurs in the cytoplasm. So, by using one strand of DNA, which is known as template strand, heterogeneous mRNA is synthesized in the nucleus. Heterogeneous mRNA has both introns and exons. Introns serve no purpose, so they are removed through a process known as splicing. Then the mRNA is added with a cap and a tail. The cap always contains a specific nucleotide known as AUG. This AUG is known as the initiation codon because the protein synthesis begins with this codon. Then a tail is added at the end of, that means at the 3 end. So AUG is added at the 5 end. At the 3 end, poly-U tail is added. That means a number of nucleotides containing uracil are added at the 3 end. So this is the tail, this is the cap. Now the synthesis of mRNA is complete and it is released into the cytoplasm. When the mRNA enters cytoplasm, the smaller unit of the ribosome gets attached to the mRNA at the 5 end. Before I proceed any further, I would like to describe ribosomes. Ribosomes are two types, 70S and 80S type. 70S type are present in prokaryotes and also in the mitochondria and chloroplast. 80S ribosomes are present in eukaryotes. Each ribosome is made of two subunits or two particles, one smaller particle, one larger particle. The subunits of the 70S ribosomes are 30S and 50S, and the subunits of 80S ribosomes are 40S and 60S. You notice that the combined weight of the two subunits will be always more than the weight of the total ribosome. Here S represents Swedberg unit. The weight of ribosomes is measured in Swedberg units. That's why the letter S is incorporated here. So, first the smaller unit comes and attaches to the mRNA at the 5 end. Then the larger unit comes and attaches. In this way, complete ribosome is assembled on mRNA at the 5 end. Now the ribosome, the larger unit of the ribosome has two sites. Actually there are 3 sites. There is another site known as E site on this side. I am not going to talk about the G site because it is not necessary. So the two sites, important sites are P site and A site. P site is also known as the peptidyl site. Here there is one enzyme, peptidyl enzyme, which forms a peptide bond between two amino acids. This is one amino acid, this is one amino acid. So here what happens is, on the ribosomes, the amino acids are joined together. Different amino acids are joined together by a peptide bond. For this peptide bond, one enzyme is required. That enzyme is present at the P site. That enzyme is peptide enzyme. That's why it is known as the P site. The second site is known as A site. A site means amino acyl site. To both the sites, tRNAs are attached. Here we have to remember one important point. Before the tRNA molecule is attached to the P site or A site, first it has to undergo amino acylation, a process known as amino acylation, and this amino acylation requires an enzyme known as amino acyl synthetase. So, first the amino acid gets attached to one ATP molecule. ATP is high energy molecule. Then the amino acid is attached to the tRNA. The whole process is known as amino acylation of tRNA. Then the tRNA are known as amino acyl tRNA. So always amino acyl tRNA gets attached to P site or A site. Initially, both P site or A site are vacant, and two tRNAs get attached to P site and A site. But later on, only this site gets vacated and tRNA comes and attaches to A site. Then, why this amino acylation process? See, peptide bond formation requires a lot of energy, and that energy is supplied by ATP. So, in this process of amino acylation, ATP is attached to amino acid and also to tRNA. That is about amino acylation. Then now let us study the structure of tRNA. This tRNA is the 3-end, this is the 5-end. tRNA molecule resembles a clover leaf. That's why it is known as clover leaf model. There are two important sites on tRNA which you must remember. One is anticodon site, this one. This is called anticodon loop. This is anticodon. Anticodon is complementary to the codons present on the mRNA. So when the anticodon finds out a suitable complementary codon, the tRNA gets attached to the mRNA. So complementarity is very important. Then this end. At the 3-end, there is one codon ACC. To this codon, amino acid is attached. So this end is known as amino acid attachment site. So this is amino acid attachment site. This is anticodon end. These two are very important. So now there are 3 steps in polypeptide synthesis. These 3 steps are initiation, elongation and termination. Now let's say this is the initiation. Now mRNA has come into the cytoplasm, and soon after it comes into the cytoplasm, what happens? The smaller unit attaches first to mRNA, then the larger unit attaches. Now both the sites, P site and A sites, are vacant. So first, one amino acyl tRNA, this is amino acyl tRNA, containing the amino acid corresponding to this one code is brought. So this tRNA has anticodon which is complementary to this codon AUG, so UAC. So this tRNA attaches here. It has amino acid attached to this end. This amino acid is methionine. So always the first amino acid incorporated into protein synthesis is methionine. Then the second site, A site, it is also vacant. Another tRNA comes and attaches here. Here the codon is CUA. So the amino acid having GAU anticodon comes and attaches here because these two codons are complementary to each other. This tRNA also brings one amino acid. So in this way, now both the sites are occupied. This is having one amino acid, this is having another amino acid. Now a peptide bond is formed between these two amino acids. After the formation of the peptide bond, the entire unit moves by one codon to the left side. So it moves like this. It jumps by one codon. And when it moves to the next codon, what happens? It comes here and this moves ahead. That means I will come here. So when it moves by one codon, what happens? This P site comes in the place of A site. A site moves to next codon, which is vacant. As P site moves to A site, what happens? The tRNA present at P site gets detached and goes back to the cytoplasm to collect the amino acid. Like this, it has moved to the next codon. So P site, which was here, first comes here, and A site, which was here, moves to this site. Now the P site, A site is vacant. So this amino acid, tRNA comes here. It has complementary codon here. This is CGG, this is GCC. So this is complementary, it gets attached. It too has one amino acid at the amino acid binding site. Now a peptide bond forms between these two. So now 3 amino acids are attached. Next, what happens? It moves by one more codon. So P comes here and A site comes here. Like that, the ribosome moves throughout the entire length of mRNA. Like this it goes. As it moves, amino acids are being added and a protein or a polypeptide is formed here. And when the ribosome encounters the termination codons, you know they are UAA, UAG, UGA. The translation process comes to an end. Now, the ribosome leaves the mRNA strand and the polypeptide is also detached from tRNA. In this way, a polypeptide chain or protein molecule is synthesized from mRNA. This is known as termination. Sometimes a number of ribosomes are attached to the mRNA and the entire structure appears like beads on a string. As the beads are present on a string, it looks like. This structure is known as polyribosome or polysome. That means a number of ribosomes get attached to the mRNA and they synthesize a number of protein molecules or polypeptide molecules. Let us say first a ribosome has attached here. As it moves along the length of mRNA, the protein or the polypeptide chain gets elongated, and by the time it comes to the end of transcription, a complete polypeptide molecule is formed and it is released. So in this way, translation takes place in eukaryotes.